But now we are moving into the second half of the short course for today. So as Alex had mentioned at the start, the CEDA resource has two components, the database on one side of the coin and then the analysis resource or the tools on the other side of the coin. So we're moving into the tools section, and I'm very happy to call Dr. Ibel Carri to the virtual stage to present the next generation tools and cancer pipelines. Thanks very much, Ibel.
Thank you, Nina, for the presentation. Can you see my slides?
Yes.
Thank you. So I'm going to present the next generation tools and how to run cancer pipelines using the NextGen site. And this is part of our extensive analysis resource that we provide in CEDAR. So in this presentation, you will learn how to access the tools to the CEDAR main page. Then we will see how to run pipelines, which are a series of bioinformatics steps that are connected one to each other. And to do so, we will see an example on how to perform neoepitope prediction. To do that, we will run three different tools. First, we will see how to run the mutated peptide generator. Then we will perform a peptide variant comparison. And finally, we will run the peptide expression annotation. Following, we will see two key features for advanced users, which are how to run a standalone versions and the tools API. And finally, I will provide you how to access to more info, documentations, and more about NextGen site. So as we will going to go through a neoepitope prediction pipeline, I want to go into the background a little bit and introduce a key concepts for neoepitopes. So tumor cells accumulate mutations, and these mutations at the DNA level can impact at the protein level as well, generating molecules that are different from what is expressed in healthy cells. Since these molecules are different, they can be good targets for the immune system. So for these neopeptides to be recognized by T cells and be called neoepitopes, two key steps need to happen. So these molecules have to be expressed, then these molecules have to be presented in the context of HLA molecules, and finally, a T cell receptor specific to recognize these molecules have to be present in the T cell repertoire and interact with the complex of the peptide MHC. So to run this pipeline, we are going to address these key steps in the immune recognition with different bioinformatic tools. So to access the tools from the CEDAR homepage, we have two different ways. We can access through the drop-down menu that says Analysis Resource, where we can see that there are T-cell, B-cells, and epitope analysis tools, and also the next generation tools. The first three links will direct you to the legacy site, which is the first version of the tools that have been developed in the IDB. And also we can access to the latest version of the tools, which is the next generation tools. And this particular website allows us the possibility to run pipelines. So I encourage you to use the NextGen site. Additionally, from the right panel, you can access to specific CEDAR tools in their most updated version. So here, for example, we can directly access the mutated peptide generator in the NextGen site. So I'm going to briefly explain what the mutated peptide generation does. So somatic mutations at the DNA level can generate different impacts at the protein level. If they fall into coding region, single nucleotide variants, insertions, and deletions can generate in-frame variants, which might alter one residue or a few residues, and frame shift variants, insertions and deletions that alter the open reading frame can generate large changes in the protein level because they alter the reading frame. So all the downstream residues will be different from the normal, and this makes them really different from serving good targets for the immune system. So the mutated peptide generation What it does is to form variant find at the DNA level. It interprets these variants and outputs a protein sequence. So the user has to provide the variants identified in the tumors, and that is made by comparing healthy tissues and the tumor cells. And these variants have to be provided in the variant calling format, which has the coordinates in the human reference genome, the reference nucleotide, and the altered nucleotide. Then the mutated peptide generator interprets these variants, performing variant annotation with SNPF. And what this does is, if the mutation falls into a coding region, then it collects all the transcripts that might be affected and imputes the type of mutation that is being generated. And then the core mutated peptide generation, what it does is to apply these mutations at the protein level and output all the mutated sequences along with the reference peptides. So we will see now an example of how to run the mutated peptide generator. So from the home page, from the next-gen site, we are going to allocate the mutated peptide generator and click on them and provide our variants in the BCF format. Here I'm going to drop my file, but you can paste the text or drop a file as well. And here, if we scroll down after the header, we can see that we have these particular fields that are critical, that contain the critical information, which are the coordinates and the nucleotides that are altered. Then I'm going to select which parameters are preferred to run. So I want to generate 9 mer peptides with the mutation located in the central position of the peptide. And I want to generate all the possible frameshift overlaps. Optionally, I can run a SNPF annotation, but since my BCF is already annotated, I'm going to skip this step right now. So now we click on Run. And we can scroll down to see the results. So we have the coordinates, the nucleotides altered. We can select how many rows we want to see in this particular view. And if we move to the right, we can see the actual peptide sequences, which is the output of the tool. We have the preference peptide and the mutated peptide with the different residue located in the middle, because we ask for the tool to generate the mutation in the middle of the peptide. So now that we have all the mutated peptides, we want to see if these peptides are good candidates to be presented in the immune system. So to do that, we are going to use the peptide variant comparison, which is a tool that compares the wild-type peptide with the mutated peptide. And as I briefly mentioned, for peptides to be presented to T cells, they have to bind to the MHC. In this figure, you can see a short peptide sequence in orange that is being presented in the context of an MHC molecule, and this is being exposed to a T cell receptor here in gray. So first, the peptide has to interact with the MHC molecule, and this happens by specific residues in the peptide, which are called anchor position, that are connected to the MHC by hydrogen bonds. And then the central positions of the peptides are exposed to being recognized by the T cell receptors. So anchor positions are super relevant for us to predict interaction with the MHC molecules and our peptide candidates. So here in this figure, I'm showing a sequence logo of the preference of an HLA molecule. Here in this case is HLA-C1502. And we can see in position 2 and position 9 that there are a few amino acids that are highly conserved, and these are the ones that are capable to be recognized and interact with the MHC molecules. So here we have a real case example that you can query in CDAR as well of a reference unmutated molecules. So here you can see that the reference in the last position have an amino acid that is not found in the motif of this particular allele. But the mutated peptide have an amino acid that is actually a part of the preference of this allele. And if we calculate the ranking with NetMHC-PAN, we can see that now the mutated peptide is among the 2% candidates of being presented in this particular MHC molecule. So this tool, NetMHC-PAN, is a really good tool to predict if peptides are likely to be presented in the context of HLA molecules. And then we can see if in the next steps if these peptides can be recognized by the immune system. So ICERFIRE is a tool that has been developed by our group in collaboration. And this tool integrates the information of binding to the MHC with further information comprising the composition of the peptides and the abundance of these peptides in cancer to predict if these peptides are good candidates to be recognized by the immune system. So we are going to run ICERFIRE to assess the capabilities of our peptide candidates to be presented in the context of the immune system. To do so, we are going to continue our pipeline. So starting from the peptides generated by the mutated peptide generator, we're going to add another step in our pipeline, which is the peptide variant comparison, and we're going to do that with unique peptides. Now you can see that the two columns, the reference and mutated peptide, are selected in blue because this will be the input of our next step in the pipeline. Different prediction models can be run to the peptide variant comparison. If you are interested in different models, you can add different prediction tools. In this case, we are going to run Neoepitope immunogenicity prediction. So we are going to select ICERFIRE for our predictions. And since NetMHC-PAN is part of ICERFIRE, we are going to remove this prediction right now, but you can choose to select to integrate different tools in this prediction. Then I'm going to select the MHC allele that I'm interested in. So for example, if I'm working with a cancer patient, I could input here all the HLA alleles that have been found in this patient. And now we select Run. If we scroll down, we're going to see the output of this tool. We can see peptide A and B that represent the wild type and mutated peptides, the allele that we have selected, and the different prediction scores that are the output of this tool. In the next-gen site, there are some columns that are displayed by default, but we can decide to include more information. So here we are going to display also the ranking predicted by NetMHC-PAN, and we are going to use the filter to select only peptides that are among the top 2%. So now we have filter and we have less candidates, and we are going to save the table to continue our pipeline. So the last step of our pipeline will be to run the peptide expression annotation. We have shown that expression is both interconnected or associated with the capabilities of peptides to be presented in HLA molecules, and also with the capabilities of them to be recognized. And actually, the level of the abundance of peptides is associated with their immunogenicity. So we are going to run peptide expression annotation, which is a tool that collects information from different public databases to tell us the abundance of our peptides. So now we add the third step in our pipeline by selecting pepX, and we are going to run that with the wild-type peptides because they are more likely to be found in the databases. There are many different databases. Now I'm going to select TCGA and with a pan-cancer approach, but if you are interested in a specific cancer type, you can select these data sets. And now we're going to run, and let's see how the output looks like. So we have our peptide and the TPM, which is the expression value across different molecules that actually contain this peptide. And we can see that among these candidates, there are a few that are highly expressed. So these are really good candidates to be immunogenic. We can export our results in a table. For example, here we have an Excel file and we can use for downstream analysis. So a few more features of the pipelines. Here in the left panel, you can access to all the parameters that have been used to run this pipeline. And if you're interested in reproducing these pipelines, you can also set a name of the pipeline and introduce your email, because if you're running a pipeline with large data set, this might take some time to run, so you will get notifications when each step is completed. Also, each pipeline is connected to a link to an URL that you can use, for example, to share your pipeline with collaborators or to continue working with this pipeline in another moment, in another time. And then if you select this link and enter to this link, you will access to the parameters that you have selected and to the data as it has been run in the first time. So here we have seen 3 CDAR tools, but we actually have a lot of tools to perform B-cell, T-cell prediction, to analyze T-cell, B-cell repertoires. And if you're interested in learning more of these tools and seeing what do we have available to perform immunology research, you can access to this drop-down menu and see all the list of tools and select the ones that are of your interest. So let's say that, for example, you have sensitive data that you don't want to upload online to the servers, or that you want to run, I don't know, a cohort of cancer patients, and you have large data sets that you want to run, and you prefer to do that in your computer. So from these tools, you can download the standalone versions that you can install in your own computer. So if you are in a company, you can get a license to be able to run these tools. And there are many different tools that can be accessed as a standalone versions. So here in this example, I'm showing you how to download the mutated peptide generator. And in this compressed file, you will find instructions to install and run these tools in your computer, in a server, in a cluster, wherever you prefer. Another option to run the tools is to use the API. So the API is an application programming interface that allows users to run jobs in the language that they prefer, not relying on the main web page. So the next-gen side API, how it works is the user first submit to our servers a prediction request. And the servers respond with a pipeline ID and a result ID. And as I mentioned before, some of these steps and the prediction steps might take some time. So you can use this ID to ask the server if our job has been finished or if it's still running. So this is the second part of how an API query looks like. So we as users submit these IDs to check if the job has been completed, and we get the status of the results of our job. So let's see how using the API actually looks like. From our pipeline, we are going to get the settings in a JSON format, which is the format that we use to submit API requests. And now we are going to the main next-gen website. Click on the API, and we are going to access to the Swagger, which is a fantastic resource that allows us to perform API queries, but also you can perform these requests in the format you prefer. So we are going to use the post method to submit a job, And here we are going to paste the parameters and the settings that we have selected for our pipeline. So here I'm pasting my parameters, and briefly you can see here that we have the information to run the 3 steps that we have selected. All the settings for the mutated peptide generator. Then we have the settings to run the peptide variant comparison, which is the step number 2 of our pipeline. And finally, we have the settings to run peptide expression annotation. So we are going to run this job. And as I briefly mentioned before, the output of, this is how the request look like. And we are going to see that we get a result ID and a pipeline ID along with further information about our particular job. So now we are going to copy our result ID to submit another request to the server, which is a get request to actually get the results of our pipeline. So now we are going to paste our ID and run execute. So our job has finished, and we can see here that we get the results of our pipeline. So for example, here is the wild type and the mutated, and one of the rows of our wild type and mutated peptides, and these are provided in JSON format. So this way of working with APIs is super flexible. You can integrate these tools in any workflow that you are interested, because this data is provided in a standardized way. If you are interested in learning more of the tools, we have extensive documentation. So here, for example, I'm going to show you the documentation of the mutated peptide generator, and this works both to use the website, or we have documentation for the standalone versions. So please get to learn more about our tools in the documentation, which is really clear and extensive. We also have a lot of YouTube videos with tutorials and more details about the database and the tools, and also all the talks that have been take place today will be uploaded to our YouTube channel. So here, for example, I want to learn more about pepX. So in our YouTube webpage, I'm looking for tools associated with videos associated with pepX. And also we have we provide all of the publications that are related to our tools. So if you go to the help and info drop-down menu, you can see that there is an option which is Cite Us, and here we provide all the papers that explain the science behind all the tools. So please feel free to contact us if you have more questions or if you are interested in using the tools. And also, if you are interested in a paid license for non-academic use, you can also contact us as well. Thank you so much.